Bismillahirrahmanirrahim. Assalamu alaikum. This is me Shamala Kadir conducting your course 317. Students, hope you all are fine. Today we have our new topic, that is glycogenesis. In the previous lecture, we covered glycogenolysis, which means lysis of glycogen or breakdown of glycogen. But here, the glycogenesis means formation of glycogen, because genesis means formation, and as it is glycogenesis, so it is the formation of glycogen. So what is the definition of glycogenesis? It is the formation of glycogen from glucose, because glycogen is the stored form of glucose. Now the sites where this process occurs, principally it occurs in liver and skeletal muscles, because it is the site where the stored form of glycogen is present. But it can also occur in every tissue to some extent. The major sites are liver and skeletal muscles. Now come towards the reaction of this synthetic pathway. The first reaction is as same as that of glycolytic pathway, where glucose is converted to glucose 6-phosphate. And the enzyme required, that is glucokinase or hexokinase. As I told you, that glucokinase is specific to liver, whereas hexokinase is present in other tissues. In this reaction, glucose 6-phosphate is formed from glucose, which required ATP, adenosine triphosphate, which will give phosphate group to glucose and form ADP, adenosine diphosphate. In presence of magnesium and enzyme glucokinase or hexokinase, glucose will convert it to glucose 6-phosphate. Now in second reaction, this glucose 6-phosphate will form glucose 1-phosphate, and this is reversible reaction. And this reaction you also saw in glycogenolysis, where glucose 1-phosphate was converted to glucose 6-phosphate in presence of enzyme phosphoglucomutase and magnesium ion. As we are going towards the glycogen synthesis, so glucose 6-phosphate is converted to glucose 1-phosphate in presence of enzyme phosphoglucomutase and magnesium ion. Actually, first intermediate glucose 1,6-biphosphate is formed, and then finally glucose 1-phosphate is formed, which is expressed in the reaction below, where glucose 6-phosphate first take phosphate group from enzyme and form glucose 1,6-biphosphate. 2 phosphate groups are added, and again it releases 1 phosphate group and form glucose 1-phosphate. So first intermediate is formed, that is glucose 1,6-biphosphate, and then glucose 1-phosphate is formed. In the third reaction, uridine diphosphate glucose is formed from glucose 1-phosphate. This required the enzyme UDP glucose pyrophosphorylase and uridine triphosphate. The form glucose 1-phosphate react with uridine triphosphate, and then it forms the active nucleotide uridine diphosphate glucose moiety and this required the enzyme UDP pyrophosphorylase. So here the UDP will form UDP glucose, and the 1-phosphate group from glucose and 3-phosphate group from uridine triphosphate entered, and 2-phosphate group are utilized in UDP glucose moiety, in the formation of UDP glucose moiety. From UDP and 2-phosphate group will release in the form of a molecule of pyrophosphate. So UDP will react with glucose 1-phosphate and form UDP glucose 2-phosphate containing moiety of glucose, that is uridine diphosphate glucose, and 1 molecule of pyrophosphate will release. So all the glucose moieties are converted to UDP glucose. Under the influence of enzyme UDP glucose pyrophosphorylase and in presence of UTP. Now this UDP glucose are ready to form 1,4-glycosidic linkage with existing glycogen molecule or to attach with primer, and that require the enzyme glycogen synthase, where carbon 1 of the activated glucose of UDP glucose forms a glycosidic linkage with carbon 4 of a terminal glucose residue of glycogen primer with liberation of UDP, because UDP will be released. But here the important thing is that there must be a pre-existing glycogen molecule. So with this, the active or the activated glucose or glucose of UDP glucose will form glycosidic bond with its 4 carbon, or there must be primer. So how the primer is formed and what is the glycogen primer and how the reaction is continued, we will see. First, it is required the formation of primer or glycogen primer is required, or there should be a pre-existing glycogen molecule. So let's see how this glycogen primer is formed, or what is this primer. Glycogen primer is formed on a protein primer known as glycogenin. If the pre-existing glycogen molecule is present, so glycogen synthase start forming 1,4-glycosidic linkage. But in case of formation of new glycogen chain, it requires the protein, that is glycogenin, which is present within the core of glycogen molecule. And it serves as an acceptor of glucose residue and making glycogen glucose complex. What happened? The tyrosine of this glycogenin has hydroxyl group. So the first glucose molecule is bound to this OH group of tyrosine of glycogenin, and the enzyme which catalyzing this reaction is protein tyrosine glucosyltransferase, and this is the intrinsic enzyme activity of glycogenin itself, where the end of glucose molecule binds with glycogenin in its reducing end. So it means it's one carbon. Whereas the non-reducing ion, that is carbon 4 of glucose, it remains free. So here you can see this, the box is basically representing the glycogen and the black part is tyrosine. And the hydroxyl group of this tyrosine is attached with UDP glucose and liberating UDP and thus form, and thus one glucose moiety is bound to glycogen first with its reducing end, and the non-reducing end, that is carbon 4, is free here. Under the enzymatic activity of glycogen, one more glucose moiety is attached through 1,4-glycosidic linkage to the previously attached glucose part of the glycogen, and thus it forms glycogen glucose complex. Here the second glucose moiety is attached with non-reducing end of the previous glucose moiety, and again the second glucose moiety release UDP and it has the free carbon 4, that is non-reducing end. So the glucose moiety come and attach, forming 1,4-glycosidic linkage. By the same mechanism, 6 more glucose units are attached, one at each time, and thus form glycogen glucose complex, which contain now 8 glucose moiety and 6 UDP more are released, because one UDP glucose will release one UDP and attach with previously attached glucose moiety. In each extension, 2 ATP molecules are expanded. How? One in the phosphorylation of glucose to form glucose 6-phosphate, as you saw, and another in the regeneration of uridine triphosphate, that is required nucleoside diphosphokinase. ATP will convert it to ADP, adenine diphosphate, releasing one phosphate group, to uridine diphosphate, that will form uridine triphosphate, and that will be ready for the utilization in the previous reaction, where glucose 1-phosphate will form UDP glucose after reaction with uridine triphosphate. Now further elongation of a straight polyglucose chain required the enzyme glycogen synthase, because after the addition of 8 molecules of glucose, the enzymatic activity of glycogen is stopped, and now further addition of glucose moiety to the previously attached glucose moieties required the enzyme glycogen synthase. As I told you, if pre-existing glycogen chain is present, so this glycogen synthase start forming 1,4-glycosidic linkage with pre-existing terminal glucose moiety. And in case of formation of new glycogen chain, after the addition of 8 glucose moiety to glycogen, the action of glycogen synthase is started, and the addition of glucose residue to a pre-existing glycogen chain occurs at the non-reducing outer end of the molecule. That is why this glycogen synthase is supposed to be the principal key enzyme which is regulating the glycogen synthesis. And when the chain has become elongated to of 11 glucose residues, now the action of branching enzyme is started, which transfer a part of alpha-1,4 chain that contain minimum 6 glucose residues. The enzyme amylo-1,4 to amylo-1,6 transglucosidase, that is the branching enzyme, it transfers a chain of glucose containing 1,4 glycosidic linkage, and the chain contains minimum length of 6 glucose moieties to the neighboring chain or the same chain to form 1,6 linkage, thus form the branch point in the molecule. So minimum 6 glucose containing chain of 1,4 glycosidic linkage, it can be 7,2, which will be transferred to another chain or the same chain and form 1,6 glycosidic linkage. Here you can see in the figure B, the cleavage between 5 and 4 glucose moieties occur, and from 5 to 11, these are the 7 glucose moieties, 1, 2, 3, 4, 5, 6, and 7. So this chain is cleaved and attached to the neighboring chain and thus form 1,6 glycosidic linkage, which is represented by here the glucose 5 of one chain and glucose 1 of other chain, and these both are linked with each other by 1,6 glycosidic linkage, thus form branch point of glycogen molecule. So the 1,4 glucosyl units under the influence of branching enzyme form glycogen molecule that contain 1,4 and 1,6 glycosidic units. The linear chain of 1,4 linkages and the branch points containing 1,6 glycosidic linkage. After the formation of one branch, further addition of 1,4 glucosyl units occur to a length of 12 to 14 glucose units, and that again required the enzyme glycogen synthase. And branch formation occurred again, that is again followed by the action of glycogen synthase for the elongation of the branches, and this process leads to the highly branched glycogen molecule formation. There are certain factors which affecting the tissue content of glycogen. Glycogenesis has a relation with glycogenolysis. If the rate of glycogenesis is more than the rate of glycogenolysis, so the tissue glycogen level will be increased because more formation and less breakdown. Now come to see factors which affect the glycogen content of tissues. First, starvation, the lack of food. This greatly decreases the liver glycogen because The stored form of glycogen goes to glycogenolysis to fulfill the requirement of glucose. And thus the glycogen content rises after an initial fall if starvation is prolonged. And this is because other route of formation of glucose is activated, like formation of glucose from non-carbohydrate compounds such as pyruvic acid and certain glucogenic amino acids. They start providing glucose to the tissues. That is why the glycogen content again rises after initial fall. But the glycogen content of muscles and brain does not fall in starvation. The glycogen content of tissue also depend on the nature of the diet. A diet which is carbohydrate containing or rich in carbohydrate greatly increase the glycogen content of the tissue, especially that of liver and the muscles. And a diet rich in proteins also increases the liver glycogen because they are also supplying glucose by other route, and after being converted to keto acid can form either glucose or glycogen, but this is occur to a lesser extent than with the carbohydrate rich diet. So the protein diet can also increase the glucose level or ultimately the glycogen content of the tissue, but it is occurred to a lesser extent as compared to carbohydrate rich diet. Now the third factor, exercise. At the time of exercise, body required carbohydrate. So this leads to decreasing the glycogen stored of both the liver and muscle because of the increased demand of the body. Another factor, tissue hypoxia. This condition also associated with decreasing the glycogen stored of liver and muscles because in absence of oxygen, no glycogen can be formed to fulfill that which get lost. So in tissue hypoxia, where no oxygen in tissue, there is rapid loss of liver and muscles glycogen. Hormones are also affecting the glycogen content, and the hormones which are involved include insulin, a very important hormone which is regulating the synthesis of glycogen because it increases the conversion of glucose to glycogen. And this is because of stimulating the glycogen synthase, which is involved in the formation of glycogen, and it also, the insulin also inhibiting the glycogen phosphorylase, which catalyze the glycogen breakdown to glucose 1-phosphate. So the insulin increasing the glycogen content of the tissue by stimulating the glycogen synthase and inhibiting the glycogen phosphorylase. The second hormone, that is adrenaline, it has opposite effect as that of insulin on phosphorylase because it stimulates the glycogen phosphorylase activity in both liver cells and in skeletal muscles. And also this hormone decreasing the activity of glycogen synthase. So ultimately it decreasing the glycogen content because adrenaline and this adrenaline increasing the breakdown of both liver and muscle glycogen because of stimulating phosphorylase and thus producing a raised blood glucose level. And also raise blood lactic acid. So the ultimate effect is decreased glycogen level in tissues. The third hormone is glucagon, and this hormone has the same effect as that of adrenaline because it also stimulates the glycogen phosphorylase. But this occurs only in liver cells, not in skeletal muscles as that of adrenaline. So this is the difference. As it increasing or stimulating the glycogen phosphorylase, so it start glycogenolysis or breakdown of glycogen in the liver or producing fall in the liver glycogen. But as it's not if produce its effect on skeletal muscle phosphorylase activity, so glucagon does not increase glycogenolysis in skeletal muscles. So only the liver glycogen breakdown started under the influence of activity of liver glycogen phosphorylase. Now, the fourth hormone, cortisol. This increasing liver and muscle glycogen level because it increasing the conversion of amino acids to glucose and glycogen. The fifth one is growth hormone. Growth hormone actually antagonizing the effect of insulin, as insulin is increasing the glycogenesis by their effect on glycogen synthase, as this hormone acts as antagonist of insulin, so it decreases the liver glycogen level and causing hyperglycemia because lysis of glycogen occur and formation is inhibited. So students, these were all about the process of glycogenesis, the glycogenesis pathway. Hope you got understood. Thank you so much. Allah Hafiz. Take care.